science. He obtained his PhD at the University of Western Brittany in France, studying and modeling the effects and bioaccumulation of paralytic shellfish toxins in the Pacific oyster. His postdoc project aims to describe the physiological effects of ocean acidification on 3 commercial bivalve species: the oyster, the sea scallop, and the Atlantic surfclam. Those results will then be integrated into bioenergetics models to enable projections on stock dynamics and economic impacts related to coastal acidification. So today's focus of his presentation will be on the Atlantic surfclam, studying coastal acidification effects upon the surfclam through an experimental and modeling approach. So we will have about 20 minutes at the end of the presentation for any questions and comments. All participants will be muted, so please ask your questions via the question box on the side there in the menu. Anthony Himes, the PhD student at the Virginia Institute of Marine Science, and he's also a Virginia Sea Grant Fellow assisting with our efforts. He'll be collating the questions to ask at the end of the presentation. So feel free to submit your questions at any time during the presentation, and they'll be moderated at the end for Emilien. So just a reminder that this webinar will be recorded and will be available on the MACAN website on the resources page in just a few days. So without further ado, we'll get the screen switched to Emilien's screen and he can begin. Thank you, Emilien.
Thank you, Grace. Thank you for the introduction, and thank you everyone for joining this webinar. Okay, I think it's good. You can see my screen now. Okay, so today, yeah, I'm going to talk about the Atlantic surfclams, and we'll try to understand how coastal acidification affect their bioenergetics. So, okay, so you know the story, carbon dioxide concentration is rising in the atmosphere, mainly because of energy production, but also because of transportation, manufacturing, agriculture, and forestry. A quarter of this emitted CO2 is actually taken up by the oceans, and that is modifying the chemistry of the oceans. So carbon dioxide reacts with water, and at the end there's a nitrogen ion that is released, and that leads to decrease the pH in the water. This decrease could reach 0.3 pH units by the end of the century, and that will correspond to a 30% increase in acidity for the 200 coming years. So another process is also decreasing the pH in the coastal areas. We call that the coastal acidification, which depend on local factors as pollution, agricultural runoff, and upwellings. So they are bringing nutrients in the coastal waters, and those nutrient will increase the primary production, so there will be more algal blooms, and when those algae will die, so after different process, they will release carbon dioxide in the water. So if we have a look to the coastal areas in the Atlantic coast of the US, so here you have, so the land areas are corresponding to, the colors are corresponding to their social vulnerability, so how their economy and social depend on the aquatic resources. So you can see that most of them are highly dependent on those resources, and the purple colors define when the seawater will reach the threshold of 7.7 pH units. And you can see that they will reach that threshold soon for the Gulf of Maine, especially the north, from 2031, during the period of 2031 to 2050. The Gulf of Maine will be from 2051 to 2070, and the states of Connecticut, Massachusetts, and New York State, those coastal water will reach that threshold in the 50 coming years. So those areas will be vulnerable to ocean acidification. And if we want to have a look at what are the biological implications of ocean acidification, we have to know what is the aragonite saturation state. So to calculate this aragonite saturation state, we just divide the concentration of carbonate in the seawater. This one will depend on the pH in the water. because as you decrease the pH, the fractional contribution of carbonate will decrease. And we divide this concentration by the saturation concentration of the carbonate. This one depends mainly on the pressure, the temperature, and the salinity. But in the mid-Atlantic bight, it is mainly the temperature changes that drive the changes in aragonite saturation state. And this is an issue because numerous marine organisms are using calcium carbonate to build shells, structures, exoskeletons. So when the aragonite saturation state, the dissolution rate will increase, and thus the balance between the precipitation and dissolution will decrease. So we know that ocean acidification affects a lot of marine organisms by modifying a lot of their living traits. But for this project, we choose to work on bivalves because they are enclosed in calcium carbonate shells that are affected by ocean acidification. And on that topic, there's a lot of studies on estuarine species, but not a lot on coastal species, such as the Atlantic surfclam. That's why we choose these species, and we'll try to understand how they are affected by coastal acidification. So briefly, the ecology of these species, we can find surfclams along the mid-Atlantic bight from Virginia up to Maine. We know that they live at deep lower, mainly at deep lower than 75 meters when the seawater temperature doesn't reach 25 degrees C. There's also some articles that show that when the temperature is varying too much, those animals, the growth of those animals will be affected negatively, and that led to observe a shift in their bathymetric distribution. So the population will move deeper to be at places where the temperature doesn't vary too much. Concerning their harvesting, so now NOAA considers that they are sustainably harvested. There's a slight decrease for the last 4 years, but the value for the Atlantic surfclams remain constant during the 40 last years. And what's going on at the place where they live for the coastal acidification? So as you can see on the picture on the right, the graph on the right. So the Gulf of Maine is has a really low aragonite saturation state between 1.5 and 1.7. The mid-Atlantic bight is more around 1.9 to 2.2. So that's the place where they are. And so as you head south, the saturation states increase. But if you look at the graph on the left, you will see that when you go deeper, this saturation state will decrease. So the surfclams are already facing like hard condition for them, and they are already facing the coastal acidification effect. So to summarize briefly, what are the effects of ocean acidification on bivalves generally? You all saw those pictures where there are larvae that are exposed to ocean acidification and shells with a lot of deformities. Actually, they are more susceptible to be affected by coastal acidification during their early stage because they are using first an amorphous calcium carbonate to build their shell, which is a polymorph that is more soluble than the aragonite that they will use later during their development. And there's also some bivalves that use a mix between aragonite and calcite that is also less soluble. Concerning the juveniles and the adults bivalves, so here this is a graph from the review of Gazo in 2013, where they summarize the effect of ocean acidification on juveniles and bivalves exposed to a low decrease in pH, 0.4. And they just counted the number of articles showing different effects on different topics. So in green, you have the positive effect, in blue, the neutral effect, and in red, the negative effects. So you'll see that the main effect concerns the calcification on the shell. So you can see here on the picture on the right, oysters that were exposed to different pH, and you can see that the one exposed to the lower pH, the color totally changed, probably because the biofilm disappeared, and also there was a dissolution of the shell. The next effects of ocean acidification on bivalves concerned the metabolism. So in this review, they identified that respiration was affected positively, neutrally, or negatively. Excretion rates were positively affected, and also some defense mechanisms were depressed, such as the immune system and the acid-base regulation. Concerning the feeding processes, they also observed a clearance decrease in the clearance rate. So the bivalve will ingest less energy when they are exposed to high CO2 concentration. And finally, concerning the mortality, they observe a negative effect, but no positive or negative effect on the somatic growth. That's probably because they only studied the effect of a low decrease in pH of 0.4. So as we can see here, ocean acidification affects a lot of different things, but we can see that the bioenergetics is affected with effect on the energy intake, effect on the maintenance costs. So we'll try to respond to our problematic by emitting two different hypotheses. So our problematic is how coastal acidification affects the surfclams energy budget. And our first hypothesis is that CO2 will affect the maintenance cost. So we define the maintenance costs are the energy required to keep body functioning at rest. So for example, we guess that the animals, the bivalves will need, the surfclams will need more energy to maintain homeostasis. And a way to estimate those maintenance costs is to measure the respiration and the excretion rates, because they are tracer for the metabolic loss. A second hypothesis that could be concomitant with the first one is that CO2 will affect the feeding processes, such as the clearance rate, which is the pumping rate of the animal to feed, and the assimilation efficiency, the ratio of organic matter that is assimilated by the animal. So now that we have those hypotheses, we need to summarize everything and try to describe what are the energetic fluxes in the surfclams. So we are working with a bioenergetics model based on the dynamic energy budget theory. So this model aims to describe the energetic fluxes within an individual and between this individual and its environment. So there is two forcing variables, the food and the temperature, that will affect the ingestion rate. This energy ingested, a part will be assimilated and then allocated to the reserve compartment. The energy from the reserve, a fraction of this energy from the reserve will be allocated to the somatic tissues. That's how an individual will grow. But before, they have to pay somatic maintenance costs. That's mandatory. And the other fraction of the energy in the reserves will be allocated to the reproduction by paying also before maturity maintenance costs. And at the end, when we sum the weight of the reserves, somatic tissues, and reproduction compartment, we can estimate the weight of the whole animal. And in this project, we will try to include the CO2 as a third forcing variable. So if hypothesis number one is validated, they will have an effect of CO2 on the somatic maintenance cost. And if hypothesis number two is validated, the CO2 will affect the assimilation efficiency and or the ingestion. So we'll need to include in this model a function that allows us to scale somatic maintenance, assimilation efficiency, and or ingestion as a function of the CO2 concentration. We choose those three parameters that are potentially affected by ocean acidification from a study of Clock in 2014. They studied the effect of ocean acidification on cockle by using an dynamic energy budget model. And from their observation, at the end, they stated that there was two different hypotheses that ocean acidification affect the assimilation efficiency and the somatic maintenance costs. So now we need data to calibrate those models and see if there is effect, what are those, and how we can quantify them. So we carried out a long-term experiment of 12 weeks where we exposed 560 individuals to 3 different CO2 concentration. So our control concentration was 566, that's the low concentration. Medium was 1380 microatmosphere, that's the medium, and 2164 for the high CO2 concentration. That corresponded to a pH of 7.80 for the control, 7.46 for the medium concentration, and 7.28 for the low, the high CO2 concentration. And just if you want to compare those results with what surfclams experience in their environment, you can see that our control condition, the organic saturation state was 1.16. And so this graph is from a survey in New Jersey's coast, and you can see that the surfclams are experiencing an organic saturation state close to the one that we have for our control. And for the medium treatment, it was 0.57 and 0.37 for the high CO2 concentration. Yeah, just to explain briefly how we managed to maintain CO2 concentration in our tanks. So we had a CO2 bottle plus compressor that were plugged to mass flow controllers, and those allowed to deliver air at different CO2 concentration. So this air was bubbled in those big white columns that you can see on these pictures, and the water was passing through those columns, and at the end they were treated, and so we at the end the seawater was treated at the different CO2 concentration. Then this water was delivered to our experimental tanks with a flow rate of 1 liter per minute, and additionally we bubbled the air at the different CO2 concentration directly in the tank to maintain the CO2. Just here, if you want to have a brief look on the chemistry of the water, you can see that the temperature and the salinity remained identical for each treatment, but that the pH, PCO2, DIC, calcite, and organic saturation state were statistically different between each treatment. And then what are the measurements that we do during the experiment? So daily we measure the algae concentration in the inflow and the outflow of each tank. We measure the temperature, the salinity, the oxygen concentration. Each 3 days we measure the water chemistry, the total suspended matter. We also sample water to identify what species of algae were present in the water. And the big part of this experiment was that every 2 weeks we sampled 33 individuals per treatment. We sacrificed them, measured their length and their weight to monitor the growth and see if ocean acidification affected their growth. We also took 7 individuals to measure their per treatment to measure their feeding rates, their respiration rates, and their excretion rates. So how did we measure feeding rates? We based on the biodeposition method. So just to briefly explain how bivalves eat. So you have seawater that is enriched with food and inorganic matter. The bivalve will pump this water, filter this water with its gills, then bring the food and the inorganic matter to his mouth, and then they will be able to sort this food and select the best food, the one with that is enriched with organic matter. They will reject a part with their pseudophysis. The other part will be ingested, assimilated, and then they will release feces. And we have an outflow that is then less concentrated in inorganic and inorganic matter. And the biodeposition method relies on the use of inorganic matter as a tracer along all that system. So from the food, the feces, the pseudofeces, and again the water, the inorganic matter in the water. So we put one individual in different small chambers. We sample their feces, the pseudofeces. We sample the incoming water in the chambers and the outgoing water from the control chambers. We filtered all of these. Then we weighted the filters, we burnt them, and then we were able to measure the inorganic and organic fraction of each of these samples. And that allowed us to calculate a lot of different feeding rates, but the main one are the clearance rate, the volume of water pumped by the animal, the selection efficiency, or they select good food compared to bad food that has a lower concentration in organic matter. The organic ingestion rate, the weight of organic matter ingested, the assimilation efficiency, the ratio of organic matter assimilated, and then the absorption rate, the weight of organic matter that is absorbed by the animal. Concerning the respiration measurement system, so we used 8 chambers. 7 were filled with one animal, another one was filled with one empty shell. We closed those chambers. We put those chambers in a thermoregulated bath. And with an oxygen meter, we were able to measure the depletion of oxygen in all of these chambers. And when we compared the rate of depletion of all these chambers with the control chambers, we're able to calculate the slope of oxygen depletion, and that's the respiration rate. For the excretion rate, so at the end of this respiration monitoring, so we just opened the chambers, recorded the time between the closing time and the opening time. We sample a few volume of water. We made it react with ammonia reagent, a reagent from an ammonia kit. We analyzed it with through spectrophotometry, and with a calibration curve, we were able to calculate the ammonia produced per an amount of time. Okay, so what are the results now? So to test the first hypothesis and see if CO2 affects the maintenance costs, here you have the respiration rate for 3 treatments. Low, the control on the left, medium on the middle, and high on the right. And you can see that the respiration rate for the control condition is lower to the ones, with the higher one being for the medium treatment. And so this curved shape relationship, it's interesting because it's exactly the same than the one that was described in Thomson in 2010. And they explained that the CO2 concentration were increasing the metabolic rate until a certain threshold, and after there was a down regulation of the energy demand. And that's exactly what we had with the surfclams. The only difference is that the concentration threshold are lower for the surfclams, so that means that they are probably more sensitive to ocean acidification than the mussels. Concerning the excretion rates, so here you can see that we had a significant difference. So the animals exposed to the higher concentration expressed a higher excretion rate. So the two, the control condition and the medium condition, were not significantly different. And we were able to calculate what we call an oxygen versus nitrogen ratio. So we just divided the energy lost through respiration and the energy lost through excretion. And this ratio normally has to be in regular condition between 16 and 20-25. That means that the catabolism of those organisms relies on the use of amino acids. That's what we had for the medium, the control and the medium treatment, 1937 and 1823. But for the high treatment, it was lower, so 11.94. And when this O versus N ratio is lower to 16, that means that the animals switch to a protein metabolism. So here we can say that the metabolism was affected and there was a switch in their metabolism when the surfclams are exposed to ocean acidification. And just to summarize those few things, here you have the metabolic energy loss, which is the sum of the energy lost through respiration plus the one lost through excretion. You can see that the control condition was different than the medium and high condition. So that allows us to validate our first hypothesis and say that CO2 affects the maintenance cost. Also, you can see that we don't have any differences in the energy loss for the medium and high treatment. It's not a linear trend that we can observe here. For the hypothesis number 2, so see if CO2 affects the feeding rates. You have here the standardized clearance rate for the animal exposed to the free treatments. Here the trend is clear. We can see that at high CO2 concentration, the animal expressed a lower clearance rate compared to the 2 other groups. This is exactly the same trend for the organic ingestion rate. So that means that those animals were eating less food, ingested less organic matter than at lower CO2 concentration. Concerning the assimilation efficiency, there's not like we can say that the mean is lower for the animal exposed to the control condition, but it's higher for the medium group. Actually, if we have a look to the selection efficiency as a function for this free concentration, we can see here that the animal exposed to the medium concentration were able to select the food better than the other one. So that could explain why the assimilation efficiency is higher for this group, because they probably selected food that was easier to assimilate. But we can't really explain why the CO2 affect this selection efficiency. So at the end, when we calculate the energy intake, so the energy absorbed by those animals for the free condition, we saw that for the control and the medium treatment, energy intake were the same, but for the high treatment, there was a significant decrease in the energy intake. So that allows us to validate also the second hypothesis, and we saw that CO2 affects the feeding rates. Okay, so now we saw that ocean acidification affect the maintenance costs, affect also the energy intake. So we are able now to summarize what is the effect on the world bioenergetics by using the measurement of the scope for growth. So on this graph, you can see that the X correspond to the energy intake for the animal of each group. The black part corresponds to the energy lost through the excretion, the gray part to the energy lost through respiration, and the remaining white part correspond to the scope for growth, which is the surplus of energy available for growth beyond that required for maintenance. And you can see here that we have a significant difference between the animals exposed to the high concentration compared to the 2 other ones. And okay, so we guess that those animals, I mean, from those results, that they will have less energy allocated to their growth. So before to pass to the growth, I just want to summarize briefly all the physiological effects that we observed during this experiment. So we saw that the energy intake was affected by carbon dioxide, especially at the high concentration. We saw the same trend for the excretion rate. We didn't observe significant result for the respiration. Maintenance costs were affected also. The control condition was different to the medium and high treatment. And at the end, the scope for growth was lower for the animal exposed to the high treatment. Okay, so now what's the effect on the growth? they were really important. So here you can see the effect, the evolution of dry flesh mass as a function of the time for the animals exposed to low CO2 concentration in black, medium CO2 concentration in yellow, and high CO2 concentration in red. Just you can see on the picture on the top right, the size of the animal at the start of this experiment and at the end. So you can see that the growth was important. And so if you compare the growth of the animal of the low and the high CO2 concentration, you can see that it was really high. So animals were really affected by CO2 concentration. Okay, so now we identified the effect of ocean acidification on the physiological rate, on the bioenergetics. We also saw the consequences on the growth. So we have everything that we needed to calibrate our depth model. So here, the dots correspond to the observation, the line corresponds to the depth model, to the standard depth model in that case. So we calibrate the standard depth model, so without integrating any effect of the ocean acidification. So that's the animal exposed to the control conditions. And we fitted the model with our observations. So here we just fitted the results, the model with the 5 4 points, because at the end, the two last one, you can see that the standard deviation increased, and it's probably because I mean we saw that animals spawned between those weeks, so we guess that the weight of those animals, we can't really trust them, and so far our model doesn't allow us to simulate the spawning, so we wanted to calibrate the model with data that we can trust. And then we did the same for the animals exposed to the medium CO2 concentration. So the only difference that we observed between the low and medium concentration were changes in maintenance costs. So we fitted the model with the data by scaling an increase in the maintenance costs effect. That's what we did, and that's that's the model prediction. The plain line, the solid line corresponds to the model prediction by including those effects on the maintenance cost. The dashed line here corresponds to just the standard model without effects. So you can see that if we don't include those effects, we are overestimating the growth. We did exactly the same for the last treatment, the high CO2 concentration. Here, between the low and the high treatment, we saw two significant differences in the energy intake and in the maintenance costs. So we kept the same maintenance cost that we estimate for the medium treatment, and we added effect on the energy intake. We calibrate our model to fit the growth in that conditions. At the end, the best fit was to decrease the feeding rates by 15%. And so you can see that here the solid line reproduced the growth. The dotted line corresponds to the growth if we include only effect on the maintenance cost. So you can see that we are overestimating the growth here, and if we use only the standard depth model, we are really far from the observations. So that's also something that validate our two hypotheses. Okay, so now we have a model that is calibrated that includes the effect of ocean acidification for those three CO2 concentration and effects on energy intake and maintenance. So we are able to make some predictions. Okay, so for those predictions, the forcing variables that we're going to use are so the free CO2 concentration that we use during our experiments. So the low concentration 566, the medium concentration 1380, and the high 2164 microatmosphere. We used the food described in the article of Monroe in 2013. The temperature showed in the article of Narvaez in 2015. So those two articles use those data from the mid-Atlantic bight where the surfclams are living. That's why we choose those articles. And also there were articles on model prediction, on surfclams growth. So there are good data to simulate surfclams growth. Assumptions here are the one that I explained just before. for the medium concentration, CO2 affects only the maintenance costs, and for the high concentration, CO2 affects the maintenance costs plus the feeding rates. The initial conditions were that we started with a 2 centimeter individual, and we started the simulation at the 1st of January, and we made the simulation during 20 years. So we just looped 20 times the food and the temperature that are shown in those graphics, those graphs. Okay, so here you can see the simulation of our model only. So here is just for the standard model, so without any effect linked to CO2. So you can see here that the growth is not too bad if we compare to observations described in the article of Weinberg in 2005. So you can see that we can reproduce the growth. Yeah, we are close to those observations. I put here the market size, which show when from our model when those animals will reach their the market size, which is 12.5 centimeter, I think. And you can see here that those animals which will reach that market size after 7 years and 8 months. So what's going on now when we simulate the growth of the animal exposed to the medium and high CO2 concentration? These are the results, and to me they are really concerning. So the dashed line corresponds to the medium treatment, and the dotted line corresponds to the low treatment. We can see that there's a huge difference in size at the end after 20 years. And if we have a look at when those animals will reach their market size, for the medium concentration, they will reach that market size 12 years later compared to the standard model. So that means after 19 years and 8 months. And for the animal exposed to the high concentration, it's even worse. During our simulation of 20 years, they never reached that size. So it is, yeah, it is concerning. But when we just saw the results in the graph that I showed you before, when we exposed our free treatment, we saw that there was an important delay. So it was just 90 days. So if you just multiply this during 20 years, at the end, you have like a difference of about 5 centimeters between the high and the middle and the medium treatment. Okay, so I'm done now with the modeling results. Just before to summarize everything, I wanted to show you some effect that we observe on the shell. So here, this is the evolution of dry shell mass as a function of the time for our surfclams exposed to the free treatment. Low in black, mid in yellow, and high in red. So you can see that there was a big effect, and at the end, the weight was really different compared to the treatment, and all along the experiment, the results were significantly different. We saw also effects like strong dissolution. So if you have a look at the picture on the right, you can see that from the hinge, there's a big hole here, like the shell totally disappeared. That makes sense because on that part of the shell, this is the shell that was built during the early stage of the animal, and they didn't use the same, they didn't use aragonite on that part, so that's why this shell disappeared firstly. So those animals were still growing, even if we observe some holes like this. But we can guess that those animals will be more sensitive to predation like this. So to conclude now, so we saw in that study that ocean acidification increased the maintenance cost. So we didn't observe, we observed a neutral effect on respiration. So we didn't have any significant results, but we saw an increase in the excretion rates. The energy intakes was also depressed by high CO2 concentration. The scope for growth was calculated, and we saw that it was lower for the animal exposed to high CO2 concentration. That led to observe an important delay in the growth rate. Also, the growth in shell mass and length was highly affected by ocean acidification, and we observed some strong dissolution event on the shell. We were able then to calibrate a model that was describing the effect of carbon dioxide on the maintenance cost and the energy intake. And by taking those effects into account, we were able to simulate the growth of these animals. And at the end, we showed concerning results by doing some simulation on clams growth under the free CO2 concentration that we experimentally studied. Just before to finish, what next now for this project? So you saw the simulation. I wanted just to validate them and confirm them by using another model, the Hoffman's models. So a model described by Eileen Hoffman. There's a slight difference compared to the DEM model. So they calculate the balance between the energy lost through respiration and the energy assimilated. And from that, they can calculate the net production. And this weight will be allocated to the weight of the animal, and that's why the surfclam will grow or lose weight. Why this model? Actually, it has been developed, calibrated, and validated and published in at least 2 different studies. Monroe 2013 and Harvey's 2015 to study different growth rate and population density. So that's a model that is already working for the surfclam, so that should be easier to use that model and calibrate it with the data that I presented today. And just before to finish, this work was really involved a lot of people in the lab, like maybe 80% of the lab here in Milford. So I'm really thankful to everyone for their help. And I want to thank you for your attention.
All right, thank you very much, Emilien, for that great talk. We do have some questions in, and anyone in the audience feel free to continue submitting questions while we ask the ones we already have. So to start, have you heard of any reports from industry members that have already noticed impacts of OA today on surfclams?
I don't think so. So we know that they changed their area of distribution following like variation in temperature. That's something that should be maybe investigated and see if they are not moving also due to ocean acidification. But no, so far I don't think we have data or just observation from fishermen.
Okay, this next question is related to some of the changes you saw in food intake. What are your thoughts on how OA will affect surfclams' preferred food sources, and could that potentially mitigate or worsen the impacts of OA that you saw in your studies?
So I think you are talking about this. So yeah, we saw that they were able to select more efficiently their food. I don't think it's gonna be a big difference that how they are able to select like better food compared to another one if their feeding rates decrease. So it's probably more a mechanism that just compensates this decrease in filtration rate. But that's just an hypothesis. We don't have any idea, and yeah, I don't think that this selection could have a big effect on their energy intake.
Okay. Our next question is, how do you think that warming and food availability may affect their growth projections?
Okay, I see. So we can't really know. So that's also something that we can include in the model. Yeah, the model take into account the temperature variability. So it will be interesting to just simulate different scenarios and see what's the effect. But we already saw that in those areas, the temperature affected negatively their growth. So even if they are moving to colder area, yeah, if the temperature increase, I don't think it could be really beneficial for them. That's just an idea, but that's something that needs to be tested, and that's probably the next thing that we're going to do with those simulations.
Okay, this next question is related to the shell changes you reported for your highest PCO2 treatment. Did you see any changes in mortality alongside those shell changes? Did any more individuals die in that treatment?
No, I think during this experiment we saw maybe 1 or 2 animals that died. So I just showed a part of this experiment. So on the total, it was more than 1,000 individuals. So two dead individuals on 1,000. It's really a few. So we didn't observe any statistical or significant mortalities for the animals exposed to the high CO2 treatment.
Okay, our next question is, will you repeat some of your experiments to find the threshold for PCO2 or pH levels that start to cause these effects?
That will be something really interesting to do. So I think that's the missing piece for the modeling. So we need to know what are the different threshold inducing changes in maintenance costs or feeding costs. But that's something I think it's hard to have like really accurate CO2 concentration in the water and be able to maintain them for a long time. And yeah, I would like to do that, but my postdoc year, it's only for 1 more year, and I don't think we're gonna have enough time to work on that. But that was my initial idea, to go further and just study just really describe the thresholds inducing that induce those effects.
Okay, our next question kind of follows from that. Do you know or have an idea of what pH level you would expect to start seeing significant impacts?
So just I want to be sure. Or 3 different treatments were 7.28, 7.46, and 7.80. I think, and I saw different articles that show that the threshold was 7.50, where we start to see like really significant effects. So we are just under that threshold. So yeah, it's probably 7.50, but I think it was with other animals. So now it will be interesting maybe just to add another treatment at 7.6 or 7.65, something around this, just to see if we have an effect and when this effect is observable.
Our next question is that your results depict averages of all surfclams in your study, but did you see any responses that became more negative over time?
I'm not sure to understand this question. Do you mean that like individually instead of taking the average?
Yeah, I think they mean kind of over the time course of your experiment. Did you notice anything that was continuing to worsen, or were they kind of stabilized at the average level that you reported?
So the thing also that we did, I'm gonna just put, this graph. So we did this experiment at 16 degrees. The food was just perfect for them. They had the maximum amount of food to them. So their metabolism was really active. And it's so they were all good at the end of the experiment. Like we didn't observe any like temporal effects on the feeding or the respiration or the excretion rates. But that's something that could be done also just to study what's the effects. So here it's like summer conditions, good food and good temperature. But it will be interesting to see what's going on at another moment in the year when the temperature is lower, the metabolism just slow down, to see if those effects just increase with the time.
Okay, I think this is our last question, unless anybody has any more that they want to drop in the question box now. Could you tell us a bit more about how you calculated the market predictions?
So the line here to say that the market size was 12.5, that's just a number that I found online. It is maybe wrong. If you have a better estimation, that will help me. But yeah, this line was just something that I found online.
Okay, thank you. I think that's all the questions we have. So with that, I'll turn it back over to you, Grace.
Great, thank you, Anthony. And Emilien, thank you so much. I really appreciate your time, and thank you for everyone attending. And again, we'll be posting this on the MACAN website in just a few days.
Thank you very much, Grace.
Thank you.